My name is Anna Blom, and I am coming from Lund University, so the local person. And it's my pleasure to introduce Claudia Kemper. And her career started in the best laboratories in the complement field, first in Hamburg and then St. Louis with Irma Gigli and John Atkinson. And then she established her own lab in King's College London. And finally, she moved back for a while, I hope only, to United States, to NIH. And she is in the Institute for the Heart, Lung, and Blood, where she is a group leader. She's also associated professor at the University of Lübeck. And I would say that Claudia is, at the moment, the most innovative researcher within the field, also slightly controversial, and I mean it as a compliment. And the complement system was discovered more than 100 years ago. It was awarded Nobel Prize in 1924. So really nobody was expecting anything really new coming out of it. But Claudia has shown us that there is a whole new world of complement within the cells. And I don't mean locally, because this is often confused, but really inside the immune cells and other types of cells where it regulates adaptive immunity, metabolism, and many other functions. So please, we are really looking forward to your presentation.
All right. Good. I would welcome everybody. I would like to start thanking the organizers, the Royal Swedish Academy of Sciences, and the Crafoord Prize Committee for inviting me. It's an honor, and I'm really, really enjoying my day here today. So I'm a funny little bird. I work with innate immunity complement, and I'm also working with T cells. And I can honestly say that the general immunologist really doesn't have much for the complement, love for the complement system. And I think part of the reason is that if you're looking at this cascade, people are often quite turned off quite quickly. But what I hope what I'm going to do today for you is that when you leave this room, that you actually find it possibly more interesting than you thought, and that you might actually look in your own work for a little bit of complement. I have to start just 2 minutes with the cascade so that you can follow me through the rest of my talk. So I think when you open the Janeway, you know, what everybody knows is complement is the system churned out by the liver, circulating in your blood as a pro-enzyme system. Key molecules are C3 and C5, and they need to be activated. They can be activated by 3 activation pathways, lectin classical alternative. They induce the formation of a C3 convertase, which then cleaves C3 into bioactive C3a and C3b. As soon as C3b formed, you generally get the formation of a C5 convertase, which then in a similar fashion cleaves C5 into C5a and C5b. And I think you all know that C5b seeds the membrane attack complex that kills bacteria, viruses, and fungi directly. What you can also see is that the other activation fragments also have functions. Basically, for almost every fragment that is activated during complement activation or generated, we have a receptor. And these receptors are expressed on immune cells. And what I did today is I listed the 3 that we are working mostly with. The anaphylatoxin receptor C3a receptor and C5a receptor, and the complement regulator and receptor CD46, which binds C3b. And it is quite clear if you don't have circulating complement or mutations in core components such as C3 and C4 or C5, you suffer from recurrent infections. So complement is really good and protective by activating the immune system and removing danger in the form of pathogens, but also self-derived danger. However, there's a really dark side to this system. Because if it's dysregulated, and it actually is dysregulated quite often, it can cause pathologic acute and chronic inflammation. And if that ensues, it contributes to a broad range of human diseases. And I have really only listed here just a handful of the diseases it contributes to. So that means we know for decades, because Anna just said the complement system was discovered over a century ago, that it is a really valid and good therapeutic target. So we have tried to therapeutically target the system, basically I would say since the 1960s. However, the outcome so far has been actually rather disappointing. So this is a snapshot from 2018, where you can see that only 4 drugs were FDA approved and in the clinic targeting complement. And today, in 2022, that is not that much more. I think it's 6 as of this morning. Now, even more importantly, the drugs that are currently in the clinic are unfortunately only targeting really rare diseases listed here down there, PNH, myasthenia gravis, or HUS. So we really are in urgent need for new complement therapeutics that target the complement system in broad human diseases such as arthritis. And I think what we have possibly done in the last 10 years with collaborators in my lab is we have possibly found a reason why we haven't been as successful in targeting it as we should have been. Because what's important to know is that traditionally complement was targeted therapeutically in circulation because we always thought it comes from the liver or it works at least in the extracellular space. And there are basically 2 slides in my presentation that I think might be take-home messages for you. This is the first one. So what we have basically found is, like Anna said, every cell in your body has a complement system that cannot just be secreted, but that is also activated and operative intracellularly. And we have this intracellular complement system set apart a little bit from the extracellular or liver-derived and have called it the complosome. And I think in a moment you know why. And what is really important is that the complosome also does something different. It controls basically metabolism, normal cell physiology, and by definition then cell survival and homeostasis and cell effector function. So the system was discovered by a really talented postdoc in my lab at King's College, Martin Kolev, in collaboration with Cassie Lezuski and John Atkinson, my mentor. I did my postdoc with John. And what you're looking at here is basically the innards of a resting human CD4 positive T cell. And what Martin found is that resting T cells, this is human, express and translate storages of C3. And that C3 is continuously cleaved, not by an intracellular convertase, but by an ancient protease, cathepsin L. And this tonic C3 activation leads to tonic C3A generation, which then signals to the C3A receptor expressed on lysosomes. And this is really something important for metabolic survival of T cells in the periphery. If you kill the intracellular C3 system in T cells, the T cells will die. So this was really a quite surprising finding, intracellular complement, and important for cell survival, meaning having a homeostatic function. And I have to be honest with you, at that time when we found that, it got really quite a bit against the grain of what we were thinking of complement as a pro-inflammatory and get-go system. However, in the next 3 to 4 minutes, I'm basically summarizing 6 or 7 years of work that we have done with collaborators, and that then showed that this system doesn't just have a homeostatic activity, but it also functions a little bit like a loaded gun. In the presence of danger, and for T cells, that's generally signified by T cell receptor engagement, the system flips to the outside, literally in seconds, and what it then does is the C3b portion engages its receptor CD46 on the surface of T cells, and this is critically required to drive interferon gamma production and thus Th1 induction in humans. This autocrine system is not required for Th2 responses, and in the lab the verdict is still out whether you need it for Th17 responses. But again, T cell or Th1 induction in humans is dependent on cell-intrinsic C3 production and CD46 engagement. Now, interestingly, this complosome or cell-intrinsic C3 CD46 axis is also important for the timely contraction of Th1 responses. CD46 also induces the co-expression of IL-10 in Th1 cells, moves them into a shutdown, memory, and tolerogenic program. So what you can see here is that the complosome is important for the life cycle of Th1 responses, homeostasis, effector function, and contraction. And what we have shown is that this is really quite important for human health. Patients that cannot express C3 or secrete it or don't have CD46 on T cell surfaces suffer from recurrent infections because they have defective Th1 responses. On the other hand, if the system is hyperactive, you have too much C3 or too much CD46 engagement in an intrinsic fashion in your T cells, Th1 hyperactivity ensures, and that contributes, we have shown, to SLE, arthritis, scleroderma, and also to T-cell-mediated chronic organ rejection. Now, importantly, and this is now something we're getting really interested in, we knew early on already that the complosome is probably amenable to pharmacological intervention. Because if we take T cells, let's say from the inflamed joint of patients with arthritis, and culture them in the presence of a cell-permeable cathepsin L inhibitor, which reduces hyperactive C3 to normal levels, we can basically normalize Th1 responses. Now, the second surprise came to us when we tried to find out how exactly is CD46 inducing Th1 responses. We performed gene array analysis of patients with CD46 deficiency T cells and healthy donors, and we were obviously expecting your normal battery of genes that lead to interferon gamma responses. However, the group of genes that was differently expressed between the CD46 sufficient and deficient T cells were to 35% metabolic genes. And then what we did together with Christoph Hess, a friend of mine, we showed that CD46 activation induces three core events. It induces the expression of genes that code for nutrient channels such as LAT1 and GLUT1 and allow the influx of glucose and amino acids into the T cell that the cell needs for its effector function. Second, CD46 induces the lysosomal assembly of the mTORC1 machinery, your hardcore metabolism machinery that integrates nutrient sensing signals. And these two events drive the very high levels of glycolysis and OXPHOS that T cells need specifically for interferon gamma production. Erica Pierce has shown that very nicely. On a third level, a third event, T cells also contain intracellular C5. CD46 increases the activation of intracellular C5, which signals to intracellular C5a receptor 1, and that induces ROS production and the assembly of a canonical NLPR3 inflammasome. This was really a surprise because the cell intrinsic IL-1 beta production did actually not kill the T cells and induce pyroptosis, but was really, really important to control the magnitude and the length of the Th1 response. So in the same fashion, CD46 also in a metabolic way or effector response induces Th1 contraction. Signals that we haven't understood yet, however, lead to cholesterol efflux, and at the same time, the expression of the MAF transcription factor, which is really important for IL-10 production. So again, the complosome basically controls human Th1 responses, induction and contraction in a metabolic fashion and in direct crosstalk with the NLPR3 inflammasome. What is really important is that the complosome, and specifically CD46, is a central metabolic hub. Complosome perturbations translate into defective Th1 responses, and they are likely amenable to pharmacological intervention. And what you're seeing here at this slide is basically one of the big issues that my lab has. CD46 is a human-specific molecule. Rodents do not express CD46. So what you're looking at here is really a human-specific pathway, and we are currently having issues to analyze this pathway in vivo because we don't have a small animal model. We made a zebrafish, has a very interesting metabolic phenotype, and we're talking with some people at the NIH to potentially do non-human primate work. That was the juncture, the last slide that I showed you when I moved my lab from King's College London to the NIH. And we started their research program with two absolutely basic questions. We wanted to understand complosome functions and complosome regulation. And one of the immediate questions was obviously what we have looked at in the last years. Is this a CD4 positive T cell specific phenomenon? Or is the complosome also present in other immune cells? And I think we used the last 3-4 years to address that question. the complosome is also important in CD8 positive T cells, where it controls interferon gamma production, granzyme B expression, and cytolytic T cell responses. Again, if you don't have a complosome in your CD8 T cells, you suffer from recurrent infections. The complosome is also present in monocytes and macrophages. With some fantastic collaborators and friends, Steve Holland, Mihalis Leonakis, and Niki Metsopoulos at NIAID, we have shown that patients that have C3 deficiency in monocytes and macrophages cannot produce IL-1 beta in sufficient amounts and suffer from recurrent infections and primary immune deficiency. And then some work with a good friend, Mariana Kaplan from Nines, showed that neutrophils also have a complosome, that the complosome in neutrophils is hyperactive, and that this contributes to SLE. And that was really a fun piece of work that we did together, because what Mariana also did is she was the first to actually look at the crosstalk between the complosome and the extracellular complement system in lupus. And what she showed is that actually both are dysregulated, and that this dysregulated crosstalk on an intracellular cellular level and serum level is actually pushing neutrophils into the hyperactive response. And her work triggered a really big question that we had in the lab. And it took us 4 years to answer that question. We asked, is the complosome also important in cardiovascular disease and sterile inflammation? And the reason why we asked that is because it is well known, 25, 30 years, that systemic complement, specifically C5a activation and C5a receptor 1 engagement on endothelial cells, is important to induce an active state of the endothelium, which allows immune cell adherence and immune cell transgression, and is a process that really is supportive in plaque formation in the endothelial wall. Second, C5a receptor activation on the surface of myeloid cells is well known to be important to induce the phagocytic uptake of antigens or particular matter, and in consequence then induce pro-inflammatory cytokine production by these danger or pump-sensing monocytes and macrophages. Also, Eike Slat's work, really beautiful, showing that myeloid cell NLPR3 inflammasome-driven IL-1 beta production is a core determinant of atherosclerotic development and possibly plaque rupture. So now I had shown you in a slide before that C5a receptor 1 is already connected to the inflammasome. So to address the question whether the intracellular complement system also plays a role in cardiovascular disease, we basically followed an orthogonal approach and engaged, because we were pretty new to the field 4 or 5 years ago, the help of some experts such as Terje Espevik from Norway University or Trondheim University, and Nathalie Nyonzima, a very, very good postdoc in Terje's lab. And we employed several new mouse models and patient samples to address this question. So this is a summary slide, because the work had been published just in December. So what we have shown is, yes, the complosome in myeloid cells is important for cardiovascular disease. The C5a receptor 1 is surprisingly expressed on mitochondria, and when the monocytes, myeloid cells, sense cholesterol crystal uptake or cholesterol accumulation, which is a hallmark of cardiovascular disease development, the C5a receptor on mitochondria is engaged, changes the direction of the electron transport chain, and via this shifts the balance from ATP production in mitochondria to ROS production. The cell engages now glycolysis to generate the energy it needs to mount a response against the sensed danger, and that leads to IL-1 beta production and the pro-inflammatory phenotype of these cells. And we have shown with the appropriate knockout mouse animals and in patient ex vivo samples that you really need to block the intracellular C5a receptor, the surface C5a receptor, and the receptor on the endothelium to block atherosclerosis. So I think we answered that question, and now know that a cooperation between the endothelial or serum complement, the immune cell-derived surface complement, and the complosome is really important to drive full-blown cardiovascular disease in humans. So at this point, we knew that perturbations of the complosome in a broad range of immune cells can contribute to human diseases. We also knew or know that the complosome is amenable to pharmacological intervention because we're working with GSK and now with Apellis now for 3 years to develop cell-permeable complement inhibitors and have shown already ex vivo that in the cases we were interested in, they can really block the C3 or C5-driven inflammatory phenotype of the cells we're looking at. Now, since we know that the balance of C3, C5 is important for cellular behavior, we're obviously also interested in tipping the balance at will. Working with an intracellular system is not easy. I think everybody who's working with STAT inhibitors or YAC inhibitors or cell cancer cells knows that. So what we decided is we were also interested in trying to find out how is the system actually regulated on the cell surface. And something really interesting happened, and it's a little bit serendipitous. When we started becoming interested in this question, we received quite a number of phone calls from people in the field who said, guys, we really can't see much of the C3 or C5 in immune cells in circulation. They really had a hard time picking up, and we knew that because it was there in low levels. And what we then thought is, okay, cells in circulation are usually not developing effector functions. The action is really in the tissue. So what we then thought is, let's just compare immune cells in the tissue versus immune cells in circulation. And we took advantage with the help of 3 key collaborators, Ben Afzali, Steve Holland, and Majid Kazemian, and compared public data from Dana Farber's lab, where she posted RNA-seq data from T cells isolated from the spleen, which we consider in circulation, and from the lung, which we consider resident. This is not as black and white as I just said it, but let's just go with this. And so we performed an unbiased analysis of the pathways that are different between the circulating and the tissue T cells. And look, complement and the coagulation cascade were the top 3 hits. And this is an unbiased analysis. This is not analyzed for complement. So what we found basically is that the defining feature of CD4 positive T cells, CD8 positive T cells, and monocytes and macrophages in tissue is the switch on of complement gene expression. And when then analyzed which of the genes are actually induced, I think you can appreciate that the whole cascade is switched on. So the word that we used, the complosome, really came true with these experiments. And now interesting, and this is published, this is why I'm just stating this, the pathway or the molecule that switches on specifically C3 in immune cells when they move into the tissue is the integrin LFA1. So LFA1 is the master inducer of C3 or complosome expression when cells move into the tissue. And what we then did is, we basically put everything that I told you here into a little bit more refined model so that we're not working on these cells in isolation or just in tissue culture plates. So what we're arguing is that every immune cell has a low-level complosome in circulation that sustains homeostasis. As soon as the cells move into the tissue in ICAM1-dependent fashion, C3 gene is switched on. We call this C3 licensing, and that can actually also, by the way, occur during the APC and T cell interaction. And once the cells move into the tissue, they integrate other environmental factors that then engage a full-blown activated complosome, which is really important for the immune cell effector function that you're looking at. Importantly, the complosome is really also important to move immune cells back into a contraction phase to prevent tissue pathology. And we're really interested in understanding the 4 life cycle phases of the complosome during an immune response. And we are very interested in the one between effector phase and contraction phase because we think if we can understand that, we might have the key to tip these responses in the tissue at will and help in autoimmunity or chronic infections or possibly even cancer. We had so far focused on immune cells. And what is now really, really exciting is that it seems that the complosome is of really broad physiological relevance. There was a landmark paper by Anna Blom's group here, where Anna showed that pancreatic beta cells also produce C3, and it works intracellularly to control autophagy. Intestinal epithelial cells rely on intracellular C1q to control mitochondrial activity and growth. lung epithelial cells, beautiful work by John Atkinson showed that lung-expressed intracellular C3 is important to control stress responses when lungs sense bacteria. Fibroblasts, really a lovely, lovely paper by the Kaufman group, are full with C3 in the inflamed joint. And the C3 in the fibroblast is increased in arthritis and contributes to the metabolic high activity state of fibroblasts and pro-inflammatory cytokine production. Kidney endothelial cells contain factor H, which controls the complosome in endothelial cells, and if this is dysregulated, it contributes to kidney injury. And in lung epithelial cells or kidney cancer cells, factor H also controls the transition from normal growth to cancer to metastasis. So what I would like to point out here is that in all of these cells, the complosome in the normal state plays a homeostatic activity, but when it's uncontrolled or perturbed or not there, it actually contributes to the diseases driven by the misbehavior of these cells. So I hope I have convinced you today that there's actually more to complement than we thought. It is much more compartmentalized than we actually appreciated in the beginning. So there's the classic extracellular complement that you all know, which is probably important to protect you against bloodborne infections. It's immune surveillance, mostly pump detection, mostly pro-inflammatory. Then you have the immune cell-secreted cell surface working complement system, which tells the cell how to behave in the presence of danger. But then you also have the intracellular complement system, which works intracellularly. And here we think it's a little bit more like what Dan said this morning. We think that this system actually senses deviations from normal cell homeostasis, metabolic, pH, and also helps to rectify that situation. And I know it's a little bit heretic, but we also favor in my lab that actually the intracellular system complement is the evolutionary older system. So we think actually the future is quite bright. Complement is really not as boring as everybody thought. It's also not fully explored, and it has really many surprises for us in store. We think that possibly thinking about the intracellular complement system might explain why the therapies that only targeted the extracellular system probably haven't worked, specifically pathologies that are tissue-bound. Vascular, eculizumab and everything works just fine because it targets the serum complement, which is important here. But anything that's tissue, you probably also have to think extracellular, local, and possibly intracellular complement. I'm finishing now with the two most important slides. One is, then I'm sent here by Bob Balaban and all of NHLVI to congratulate you on the Crafoord Prize in Polyarthritis 2021. You're probably a academic clinician's dream with what you have achieved for science, medicine, and the patients. So thank you for that and congratulations. And I just, the other two, I just finished here really with the names and few of the faces of the people that did all the work of the journey that I have shown you, that, you know, yeah, of the data that I have shared with you today. So thank you very much.